Good morning, my dear students. Welcome to our Zoology online class. I hope you are very happy. In the last class, we studied about three topics, students. The first topic is MTP, medical termination of pregnancy, and the second topic we studied about is sexually transmitted diseases, and the third topic is cervical cancer. Okay, so I hope you all studied all these three topics. Okay, so here MTP meaning, once again I repeat the definition for this MTP. Okay, MTP meaning the medical termination of pregnancy by using a non-surgical method is called MTP. Then the second topic is sexually transmitted diseases. Diseases which are transmitted from one person to another person through sexual contact is called sexually transmitted diseases. And the third one is cervical cancer. It's a disease. Okay, it's also a sexually transmitted disease affecting the cervix region of the female reproductive system. So understand. So we have studied in the last class, we have studied about these three topics, and already many times I told you some 5 mark questions, 2 mark questions, and many 1 mark questions also there. Okay. Shall we move to the today's topic? Okay, today we are going to study about some more topic. So today we can complete this third chapter also. Already we have completed the first 2 chapters. Today we can complete this third chapter. Okay, come to the topic. See, today the first topic is infertility. Our first topic is infertility. See, infertility is one of the major problem nowadays. This infertility is one of the major problem affecting both male and female. Okay, so the male and female both type human beings also affected by this infertility problem. Understand? So if any male is affected with this infertility problem, it is called male infertility, and if the female is affected, it is called female infertility problem. Okay, so the meaning for this male infertility is if a man is unable to produce sufficient number of sperm, or if a man is unable to produce good quality sperm, is called male infertility. So once I repeat, if a man is not or unable to produce sufficient numbers of sperm, or a man is unable to produce good quality sperm, is called male infertility. And female infertility meaning if a woman is unable to become pregnant or unable to maintain pregnancy is called female infertility. Okay, inability of the woman to become pregnant or inability of the woman to maintain pregnancy is called female fertility. Okay, so female infertility. So today we are going to study about the first topic is infertility. And already I told you this infertility is a major problem affecting both sex, both male and female. Understand? So now we are going to study about the reasons or the causes for this infertility problem. Listen very carefully. See, here I have written all the reasons or all the causes for the infertility problems. So now we are going to study about all these reasons one by one. The first reason is tumor. Tumor meaning in Tamil we can say kattigal. So because of some reason, some tumors are developed in the pituitary gland or the reproductive organs. Understand? So if any tumor occur in the pituitary gland or inside the reproductive organs, it leads to infertility problem. So this is the first reason. Then the second reason is mutation. See, genes is one of the very important factors present in each and every cells. Okay, the functions of the cells. So some characters are controlled by the genes. Okay, so here you all know in both male and female reproductive system, some sex hormones also produced. The male sex hormones produced by the male reproductive system, female sex hormones are produced by the female reproductive system. So some genes only are responsible for the production of these sex hormones. Okay, sometimes the genes responsible for the production of this hormone is involved in mutation. Mutation meaning sudden change. So the genes responsible for the production of sex hormone is involved in mutation or sudden change. So it leads to infertility. So the mutation in the genes responsible for the production of sex hormone is the second reason for infertility. Then the third reason, malformation. Some sex organs are, in some person's body, the sex organs or the reproductive organs are not formed properly. Particularly the cervix region and the fallopian tube. Okay. So in female reproductive system, if the cervix or the fallopian tube is not formed or not developed properly, it leads to infertility. So this is the third reason. Then the fourth reason is stress. If a person is continuously affected by stress, over stress, it leads to infertility. In female, because of this stress problem, over stress, it leads to some problem in the menstrual cycle also. If any problems occur in the menstrual cycle, it also leads to infertility. So, over stress is the next reason or one of the causes for the infertility. Then toxin, intaking of some toxin. Unknowingly, nowadays we are eating different types of food materials. Some food materials consist of some chemical-like substance, chemical substance. So when we continuously use these chemicals, it become a toxins. Okay, it may be act as a toxin inside our body. So it is also one of the reason for the infertility. Then the next reason is using alcohol, tobacco and marijuana. Here marijuana meaning is an illegal drug. So when we use these unwanted drugs continuously or unwanted substance continuously, it leads to infertility. Then next reason is injuries of gonads. Gonads, you all know the meaning of the word gonad. Male gonad is called testis and the female gonad is called ovary. So if any injuries occur in the testis or ovary, it leads to infertility. Then next one is PID. See, this is a disorder or disease affecting the female. Pelvic inflammatory disease. Okay, this disease maximum affect the female. Then uterine fibroid. This disease also affecting the female. Then endometriosis. This region or this disease also affecting the female. Understand? So, pelvic inflammatory disease or the uterine fibroids or endometriosis. So, if any female or woman is affected by this disease, it leads to infertility. Then the next reason is low body fat. Some people, particularly women, okay, some girls feel that when we eat more amount of food, it leads to over body weight. So because of this fearing, the people are not eating properly. So if the amount of fat is reduced in the body, it leads to infertility. So because of the fearing about gaining of overweight, the female or the young females are not eating properly. So the amount of fat is reduced in the body, it leads to infertility. Then next one is undescended testes. Undescended testes. See, undescended testes means, already I explained, the testes, both the testes in female reproductive system, both the testes are kept inside a bag-like structure. It is called a scrotum, and the scrotum is always hanging below the abdominal region for one important purpose. Because the scrotum or the testes should maintain 2 to 3 degrees lower temperature than the normal body temperature. Then only the testes can produce very effective sperms. Okay, so for this purpose only, the testis is always hanging outside the body inside a scrotum. Understand? In some male reproductive system, the testis is not hanging outside. Okay, it is located inside. Okay, so this is one of the reason. And so this condition is called undescended testis. So testis should hanging outside the body, inside the scrotum. Okay. So in some person's body, the testis is not hanging outside. Okay. So this condition is called undescended testis. So this is also one of the reason for the infertility. Then tight dress. This infertility is affecting. This reason maximum the male. Okay, gents only affected because of this. But a tight dress. So when people using very tight dress, particularly the man, particularly male, okay, wearing the tight dress, when they wear tight dress, the temperature of the scrotum is increased. Okay, temperature of the scrotum is increased. If the temperature of the scrotum is increased, automatically the sperm production is affected. Okay, so we have to avoid using tight dress. So tight cloth is one of the reason for male infertility. Understand? Then next one is underdeveloped ovaries and testes. Ovaries is the female primary sex organ, testis is the male primary sex organ. So if this ovary or the testis is not developed properly, this is one of the reason for infertility. Then next one is antibody. See, in some female body, the female body producing some antibodies. These antibodies acting against the male sperms, against the sperm of her partner. So, antibodies produced inside the female body, but these antibodies act against her partners or act against the male sperm. So, this is one of the reasons for the female infertility. And the last one is autoimmune response. Okay, here autoimmune response meaning in some male's body, normally the immune system, the cells involved in the immune system, the main functions of these cells is destroying the foreign particles. The cells present in the immune system only destroying the foreign particles. Sometimes in male body, the immune cells destroying their own sperms. Okay, in male body, the immune cells destroying their own sperms. Understand? So this is called what? Autoimmune response. So this is also one of the reason for male infertility. Understand, my dear students? So I hope you all understood all the reasons. So it's a 5-mark question. Okay, this is a 5-mark question. So once again repeat all the reasons for both male and female infertility. The first reason is tumor. Maximum if the tumor is occur in the pituitary gland or the reproductive organ, it leads to infertility. The next one is mutation. Okay, if the genes responsible for the sex hormones production is sometimes involved in the mutation, so it leads to infertility. The next one is malformation. If the reproductive organs, particularly if the cervix or the fallopian tube is not developed, not formed properly, it leads to infertility. Then overstress, particularly female body. If the female is continuously affected by stress, overstress, it leads to some problem in the menstrual cycle, so it leads to infertility. Then toxins, okay, in taking up some toxins. The next, alcohol, tobacco, then marijuana. So these drugs, when we use these drugs continuously for more time, it leads to infertility. Then injuries of the gonads. Gonads meaning reproductive organ. Male gonad is called testis and the female gonad is called ovary. So if any injuries occur in the gonads, it leads to infertility. Then diseases. These 3 diseases affecting the female. Pelvic inflammatory disease, uterine fibroids, then endometriosis. If any female is affected with these disorders, it leads to infertility. Then low body fat. Okay. So suppose because of fear, some female are not eating properly, not eating the fat-rich food items. So it leads to infertility. Then undescended testis. In some male reproductive system, the testis is not hanging outside properly, so it leads to infertility. Then tight dress. Okay, this is a problem in the male. Okay, so if a man is wearing very tight dress, automatically the temperature of the scrotum is increased, so it leads to infertility. Then underdeveloped ovaries and testes. Ovaries and testes, both of the primary sex organs. So if these 2 primary sex organs are not developed properly, it leads to infertility. Then antibodies. In female body, some antibodies are produced. These antibodies act against her partner's sperm. Then next, autoimmune response. In male body, the cells present in the immune system destroying their own sperm cell. Understand? So these are the reason for the infertility or causes for the infertility. So I hope you all understood this topic. Understand? Okay. Okay. Now we are going to move to the next topic. ART. See, yeah, nowadays this infertility is one of the major problem. So to prevent this infertility problem, some techniques, some methods are used in the medical field nowadays. Okay, so these methods are togetherly called ART. Okay, ART. See, so there are number of methods used to maintain pregnancy, are used to handle the both sex cells, okay, outside the body. Okay, number of techniques, the collection of procedure used to handle the sperms and egg or zygote outside the body to achieve a successful pregnancy is called ART. So there are nowadays, I already told you, there are many methods are used in the medical field nowadays. See, the first one is IVF, in vitro fertilization. IVF meaning in vitro fertilization. See, in this method, in this in vitro fertilization method, both male sex cell and the female sex cell, both sex cells are allowed to join outside the body. That means, so inside in a laboratory, inside a culture vessels, both sex cells and the sperm cells are allowed to fuse, join. Okay, so after allowing these two sex cells to fuse, zygote is formed. This zygote is transferred to the mother's uterus. Inside the uterus, this zygote is developing as a baby or fetus. So here in vitro fertilization meaning the fusion of both sex cells, both egg and sperm cells outside the body or in a laboratory is called in vitro fertilization. And once upon a time, this in vitro fertilization technique is used to treat some women. Okay, the women those who are affected with damages in the fallopian tube, blocked fallopian tube, or absence of fallopian tube. So for these cases only, this in vitro fertilization process is used once upon a time. But nowadays, maximum for all the infertility problem, this in vitro fertilization is used in the medical field. Okay, then this in vitro fertilization consists of some steps. The first step is called ovarian stimulation. Next, egg retrieval. Third step, fertilization. Fourth step, culturing of embryo. Then the last one is transfer of embryo. So these are some of the steps involved in the in vitro fertilization. Here, to do this in vitro fertilization process, first one hormone is injected in the female body. Name of that hormone is called human gonadotrophic hormone. So first human gonadotrophic hormone is injected in the female body. Immediately the ovary is stimulated. So after 34 to 37 hours of this injection of this hormone, the egg is collected from the ovary of the mother. Once I repeat this point. So in vitro fertilization, the first step is human chorionic gonadotrophic hormone is injected in the female body. After 34 to 37 hours, the egg is collected from the ovary by giving a small anesthesia. Here anesthesia meaning in Tamil we can say Mayakamarand. Okay, so first the human chorionic gonadotrophic hormone is injected in the female body. Now the ovary is stimulated. So after 34 to 37 hours, the egg is collected from the ovary. Then the unwanted, the surrounding tissues, everything are removed from the egg. Okay, removed from the egg. Now this egg is kept in a culture vessel. Now the egg is kept inside a culture vessel. Now the sperm is added inside the culture vessel. The sperms are added inside the culture vessel. So for this in vitro fertilization, we have to add minimum 10,000, maximum 1 lakh sperms. So inside the culture vessel, only one egg only there. But we have to add 10,000 to 1 lakh sperm inside this culture vessel. Finally, one sperm will enter inside the egg and the egg is now converted into fertilized egg. Now the egg is converted into fertilized egg. So after now the fertilization process is over. Now the egg is transferred. Okay. Now the egg is transferred to the uterus of the mother. to the uterus of the mother. So inside, sorry, it is transferred to the uterus of the mother. Inside the uterus, it is developed as a baby. Understand? Then, if we need more eggs, sometimes it will not, this method will not give success. So we have to inject or implant more eggs inside the uterus. So for this purpose, we have to collect more egg from the ovaries. Okay? So one by one, all the eggs are transferred to the uterus. Finally, we will get one success result. And the excessive embryos or the excessive eggs are stored for future use. Okay, by using a technique, it is called cryopreservation. So by using cryopreservation technique, the excessive eggs are stored for future use. Understand, my dear students? So that's all about this first one, in vitro fertilization. The next one is called IUI. See, intrauterine insemination. Intrauterine insemination. See, intrauterine insemination is a second ERT tester technique. See, in this technique also, the ovary is stimulated. For this purpose, we have to inject one hormone. The hormone's name is all know, human chorionic gonadotrophic hormone. So, in this method also, first human chorionic gonadotrophic hormone is injected in the female body. Now the ovaries are stimulated and the ovaries are ready to produce more eggs. Understand? Understand you? Now, the sperm or the male sex cell is collected from the husband or from another one donor, male donor. Okay, so the sperm cells are collected from the husband or from anyone, male donor. Understand? Now the sperms is inserted inside the uterus. Okay, so it's a ovary, uterus, fallopian tube. Ovary, uterus, fallopian tube. It's a uterus. Okay, now this region is vaginal region. Okay, so now the sperm collected from the donor male is inserted inside the uterus. Okay, already from the ovary, the ovaries are stimulated, the ovary is stimulated and the ovum is produced from the ovary and it is inside the fallopian tube. Now the sperm is moving to the fallopian tube and the fertilization process takes place. After the fertilization, the fertilized egg or the zygote is moving to the uterus and implanted in the uterus wall. So this method is called intrauterine insemination. Intrauterine insemination meaning the male sex cells are collected from the husband or from a male donor and it is inserted or transferred to the uterus of the mother or uterus of the female. Then next one is called zygote intrafallopian transfer. Zygote intrafallopian transfer. Listen very carefully. Zygote intrafallopian transfer. In this method, the egg cell, so see, zygote intrafallopian transfer. 8-cell stage zygote or 8 plastomere stage egg cells are transferred directly to the fallopian tube. Directly to the fallopian tube by using a laparoscope. So by using a laparoscope, the 8-cell stage zygote or 8 plastomere stage zygote is transferred to the fallopian tube. So this method is called zygote intrafallopian transfer. Understand? After transferring this 8 plastomere stage cell inside this fallopian tube, the 8 cells are increasing in their number. 16, 32, then 64, like that, the number is increasing. Then slowly this zygote is attached to the, moving to the uterus region and it is implanted and developing as a baby or fetus. Understand up you? So zygote intrafallopian tube. Okay, so 8-cell, very important point, 8-cell stage plastomere or 8-cell stage zygote is transferred to the fallopian tube directly. Understand up you? Then IUT. IUT, intrauterine transfer. IUT meaning intrauterine transfer. See, in this method, more than 8 cell stage plasmid or zygote is transferred to the uterus region directly. So the difference is here the 8 cell stage zygote is transferred to the fallopian tube, but in intrauterine transfer the more than 8 cell stage plasmid is the zygote is transferred to the uterus. Okay, to the uterus. Understand all of you? Then next, so now we have completed 1, 2, 3, 4 important ERT methods. Understand? Then next one is GIFT. Okay, gamete intrafallopian transfer. Gamete intrafallopian transfer. See, gamete fallopian. So gamete meaning ovum. Male gamete is called sperm and the female gamete is called egg or ovum. So the ovum is collected from one female and the sperm is collected from another one male, both sections. Now the egg and the sperm, or the ovum and the sperm, both are directly transferred to the fallopian tube. Okay, so gametes are directly transferred to the fallopian tube. So just imagine the ovum is transferred here, the sperms also transferred here. Now the fertilization process takes place here. After fertilization process takes place, zygote is formed, then the zygote is slowly moving to the fallopian tube and it is implanted in the uterus. Sorry, moving to the uterus and it is implanted in the uterus, wall of the uterus. Understand? So gamete intrafallopian transfer. Then next one is ICSI. ICSI meaning intracytoplasmic sperm injection. Okay, intracytoplasmic sperm injection. In this method, okay, just imagine it's a egg. Okay, it's a egg. One sperm. Okay, one sperm is directly injected into the cytoplasm region of the egg through the focal point. So just imagine it's a focal point. So through this point, the single sperm is inserted or transferred to the cytoplasm of this egg cell. Immediately the egg cell is converted into fertilized egg. The egg cells are converted into fertilized egg. Understand of you? So intracytoplasmic sperm injection. So in this method, one sperm is injected into the cytoplasm of the egg through the focal point very carefully. Through the focal point very carefully. Understand of you? And maximum 75 to 85% of this method is giving successful rate. Understand of you? So listen very carefully. ICSI meaning intracytoplasmic sperm injection. So in this method, only one sperm is injected into the cytoplasm of the egg through the focal point very carefully. And this method giving 75 to 85% success rate. Understand? So after the fertilization process is over, it is transferred to the uterus. So inside the uterus it is implanted and it is developing as a baby. Understand of you? Then next, listen very carefully. Surrogacy. Okay, see, surrogacy means in Tamil we can say vadahaitai. Okay, see, listen. It's an agreement. Surrogacy meaning is an agreement given by one woman. Okay, given by one woman to carry the pregnancy for another parent. Okay, carry the pregnancy for another one woman. Okay, who will become the parent for the newborn baby. Listen very carefully. Surrogacy meaning is agreement given by one woman to carry the pregnancy for another one woman who will become the parent for the newborn baby. See, just imagine. Okay, see, one woman, one man. Okay, see. Now, surrogacy means this this woman is a surrogate. So this woman giving one agreement to this woman. She is ready to carry the pregnancy for this woman. Okay, she is ready to carry the pregnancy for this woman. So after the pregnancy pregnancy is over, child is delivered from this mother's body. Okay, from this lady's body. So this female giving the baby to these parents. Okay, now they are the parents for the newborn baby. So for in this method, okay, so for this method, ovum is collected from this woman and the sperm is collected from this man. Okay, so this is a wife husband. Okay, the ovum is collected from this woman and the sperm is collected from this man. Now both ovum and eggs are fused outside the body or jointed outside the body. So fertilization process takes place outside the body. So after the fertilization process is over, this egg cell is converted into fertilized egg. The egg cell is converted into fertilized egg. Now this fertilized egg is transferred to the surrogate mother. This fertilized egg is transferred to the uterus of this mother. So the baby is developing inside the uterus of this mother. So after 9 months, the baby is delivered by this lady, okay, this female. But the parent is these two people. So this is called surrogacy. This is called surrogacy. So once again, I repeat the meaning. This surrogacy. Surrogacy meaning is agreement given by one female. Okay, agreement given by one female who is ready to carry the pregnancy for another one female. Okay, here the another female is the parent for the newborn baby. So it is called surrogacy. It is called surrogacy. I hope you all understood all these ART methods. Understand all of you? So listen very carefully, dear students. Here this in vitro fertilization is little big, so it's a 5-mark question. All the other questions are, all the other methods are 2-mark or 3-mark question. Okay, 2-mark or 3-mark question. And one very important 1-mark question is here. Here 8 cell stage blastomere, here more than 8 cell blastomere. This is the difference. Understand of you? So it's a 5-mark question in vitro fertilization. All these other methods are 2-mark or maybe asked for 2-mark or 3-mark questions. Understand of you? So today we have completed 2 important topics. The first one is already I told you about infertility. Okay, the infertility and the reasons or the causes for infertility, and the next one is called ART. ART. Okay, then a small 2-mark question. Amniocentesis and chorionic villus sampling. See, CVS. See, these two are the tests. These two techniques are used to find out the abnormalities in the chromosome of the fetus. When the fetus is developing inside the uterus of the mother, sometimes the fetus may be affected with any problems. Okay, so the chromosomal abnormalities of the fetus is identified by using these two techniques. Here amniocentesis, in this method, through the abdomen, a small needle is injected inside the amnion. Amnion means it's an extra embryonic membrane. Already we have studied the structure of uterus. Okay, so when the baby is developing inside the uterus is covered with a membrane. It is called amnion. So inside the amnion, one fluid is there. It is called amniotic fluid. So through the abdominal region, a small needle is injected inside the amnion, and the fluid, a small amount of amniotic fluid is taken outside. And this amniotic fluid consists of more amount of fetus cells. The cells of the fetus is highly available in the amniotic fluid. So a small amount of this amniotic fluid is taken from the amnion and it is tested. So when we check these cells, we can identify if there any abnormality is there. If any chromosomal abnormality is there in the fetus, we can easily identify. Does not you understand? Then Next one is called chorionic villus sampling. Okay, so chorionic villus sampling means this is also the same technique used to identify the chromosomal abnormalities of the fetus. Okay, but here the tissues are taken from the placenta. Tissues are taken from the placenta. Understand, my dear student? So these two techniques are used to identify the chromosomal abnormalities of the fetus. Chromosomal abnormalities of the fetus. Understand? Then next one device. Fetoscope. 2 mark question. Fetoscope. See, this is a device used to hear the heartbeat sound of the fetus. When the fetus is developing inside the uterus of the mother, we can hear the heartbeat, okay, of the fetus by using a device. It is called fetoscope. It is called fetoscope. Normally, the adult person's heartbeat rate is 72 to 80 beat per minute. You all know that. But the heartbeat rate of the fetus is 120 to 160 beats per minute. So the heartbeat rate of the fetus is more than the adult person. So 120 to 160 times the heart is beating inside the fetus body. Understand? So if any abnormality is there, if the heartbeat number is increased or decreased, it indicates the fetus is not receiving the sufficient amount of oxygen, or it indicates some other problem to the fetus. Understand a few? So this heartbeat sound is checked by the doctors whenever the pregnant lady is going to the hospital. And as a final stage during the labor time, labor, 2 mark question already we have studied in the last chapter. Labor meaning a series of events that helps to expel the baby outside from the uterus is called labors. So at the time of delivery, during the time of delivery, the heartbeat is continuously monitored by the doctors. Understand a few? So I would say repeat this fetus about this fetoscope. See, fetoscope is a device used to hear the heartbeat of the fetus. Understand? Normally the heartbeat rate of the fetus is 120 to 160 beat per minute. If any variations occur in this range, it indicates the fetus is not receiving sufficient amount of oxygen or the fetus is affected with some other problem. Understand? And this heartbeat rate is continuously monitored during the time of delivery. Understand, my dear students? So now we have completed many important topics and now we completed the 3rd chapter also. Okay, hope you all understood today's topic. So today 2 important 5 mark questions. Okay, the first question is infertility and the causes for infertility, and the second question is in vitro fertilization. These 2 are the 2 5 mark questions. Then all these methods are otherwise called, all these methods may be asked for 2 mark or 3 mark. Here intrauterine insemination, zygote intrafallopian transfer, intrauterine transfer, gamete intrafallopian transfer, intracytoplasmic sperm injection, then surrogacy. The surrogacy is a very important 2 mark question. Understand a few? So the last class also many students completed that portion. And some of them, many of you sent the answer to me. I corrected that answers and I told them what are the mistakes you mentioned there. So today also all study these question answers and take photo and send to me my WhatsApp number. Okay, immediately I will correct and say the mistakes. Okay, so if you forgot to write any points, I will repeat that points. Okay, so study well. All these questions are very important. 2 mark, many 2 mark, 5 mark and 1 mark questions also they are study well. Okay, we will meet in the next class. Thank you.